Good afternoon. Thanks for joining us for today's webinar, Disease Control with DMI Fungicides, as part of a resistance management program presented by PBI-Gordon and taught by Brian Aynardi, Ph.D. My name is Lisa Wick. I'm the Senior Manager of E-Learning Programs here at GCSAA and will serve as today's moderator. A couple of quick housekeeping notes before I turn things over to Brian. We are recording today's event. You'll receive access to that recording as part of the follow-up email. Your audio is muted in this system. We encourage you, though, to ask questions as we go along. You can do that by typing into the question answer box on the control panel. If your control panel is minimized, you'll still see a little narrow strip with an orange rectangle at the top. Click on that, and that will maximize the control panel. And you'll see the question answer box. You'll also see the handout area. We have a couple of handouts that you can download there if you'd like to take notes as we go along, so you can go ahead and do that. Today's session is eligible for 0.1 GCSA external education points. At the end of the program, I'll read a code that you can use in the association's website. We hope you find today's webinar valuable. It's brought to us in partnership with PBI-Gordon, with a full line of herbicides, insecticides, fungicides, growth regulators, and other products. PBI-Gordon Corporation is a national leader in the professional turf and ornamental management industry. Brian Aynardi, Ph.D., is the Northeast R&D Scientist and Manager of University and Contract Research for PBI-Gordon Corporation. He earned both his bachelor's in turf science and his Ph.D. in plant pathology at Penn State University. Aynardi worked as a research technician at Penn State prior to joining the team at PBI-Gordon and resides in State College, Pennsylvania. Please join me in welcoming today's presenter, Brian Aynardi, Ph.D. Brian?
Okay, thank you, Lisa, and welcome everybody to the webinar. I'll turn my webcam off here in a second. I figured to just, any of you I haven't met before, put a face with the name so you know who I am. But today we're going to talk about fungicides as a part of a resistance management program. I have a lot of information here from some basic level plant pathology things, getting into really specific things with DMI fungicides, and then also moving from that into more of the practical side. So you know how to best use DMI fungicides, maybe where to best use them, potential concerns, and also, of course, as the title indicates, how to use them as part of a resistance management program to get the most out of your pathogen control. So with that, we'll go ahead and get started. The first thing I wanted to discuss, and I talk about this in most talks that I do, is really some basic plant pathology. First, what is a plant disease? Okay, well, plant disease is the visual result of a dynamic, sustained interaction between a susceptible host, that's really important, a virulent pathogen, which is mediated by conducive environmental conditions. These three components make up what we in plant pathology call the disease triangle. Pathogens of plants, these include fungi, bacteria, viruses, and nematodes. For the purposes of today's talk, we're mostly going to talk about fungi. There's also abiotic stresses. Some folks can consider these to be diseases as well. But overwhelmingly, diseases of turfgrass are going to be caused by fungi. Also, it's important to note that what you see is the disease. That's the physiological response. What causes a disease is the pathogen. So what we're actually controlling, there's a lot of loose terminology in our industry, but what we're actually doing is controlling the pathogen. We're not really controlling the disease. So signs of a pathogen are visible structures that are readily observable on affected plants. Examples of these would be things like aerial mycelia that you would see with the Pythium or the dollar spot pathogens, sclerotia, like you would see with the gray snow mold pathogen, or mushrooms, which you see with a number of different fungi, but most commonly with fairy ring. Symptoms are the physiological response of a host plant due to an attack from a pathogen. Examples of these would be things like lesions, chlorosis, patches, blight, necrosis, things of that nature. So what are some signs of a pathogen? Here are some sclerotia you see. This is from the gray snow mold pathogen. Here are acerbuli that you would see with the pathogen that causes anthracnose. Puffball or mushroom that you see with the fairy ring pathogens. Aerial mycelia, as you see, with a Pythium pathogen, and then of course red thread and pink patch, which typically occur together. You see these flocks of aerial mycelium. So those are signs, right? That's where you're actually seeing the pathogen. Symptoms of a disease are going to be things like your brown patch rings that you see, or patches, lesions on a particular plant. With this one, those of you out there guessing what it is right now, these are lesions from the dollar spot pathogen. Again, you have dollar spot and brown patch coexisting in the same place, and here's going to be some pathogen, or I'm sorry, some symptoms of Pythium blight. I do want to let you know, if you're ever curious when you're looking at stuff as to whether or not it's a disease, one thing that's usually a dead giveaway is if you see some level coming out from the center. So fungi move out radially or in a circle. If I go back here, a few will show you with brown patch. So they most always move out radially in the field, or at least where they're affecting turfgrass plants. But a lot of times what you see with disease is you see dead turf in the middle, or mostly dying turf. And as you move out, you tend to get less and less symptoms on individual plants. That's typical of a disease. So if you see just a big dead square in your backyard and you happen to have a party the night before, and it's because a pool was sitting there, well, that's because it was from the pool. If you see an area of concentrated death moving out to less death, that's where you would get a disease, or that's symptomatic of or typical of a pathogenic attack. So there are two different things that are occurring when we talk about fungi. Okay, there's true fungi and there's fungal-like organisms. Fungal-like organisms would be your oomycetes. Okay, and here for those of you who are guessing, this would be these are oospores in root tissue. All these round double-walled structures. And oomycetes are not true fungi. This is actually really important from a management perspective, because you want to know whether you're controlling a true fungi or a real fungus, or an oomycete, which it's not a true fungi. A lot of people like to call these water molds. They do produce mycelia in spores, which are very similar to true fungi. If you're ever looking under a microscope, one specific differentiation would be that the mycelia, or the hyphae in particular, of oomycetes do not have septation or cross walls in their mycelia. So that's important. Another important distinction is that oomycetes lack chitin in their cell walls. They don't form ergosterol. So ergosterol is very similar in the cellular membrane for structure, much like humans have cholesterol. True fungi have ergosterol, but oomycetes do not. Okay, and I have this bolded because we'll get to this part a little bit later on in the talk. Pythium species reproduce sexually from oospores, which survive in the soil and plant debris. And because of the production of oospores and zoospores, which are motile spores that move with water, it has a high potential for resistance to develop. Oomycetes are really tough to control, and these are actually the type of organisms that caused the Irish potato famine, which was caused by Phytophthora infestans. And I know last time I gave a presentation, Lisa was happy to learn this because she's from Irish descent. So those of you who are Irish and you wonder how you came to the United States, it's probably because you came in the mid 1800s during the Irish potato famine, which was caused by an oomycete. True fungi, there's two different types of true fungi. There's ascomycetes and basidiomycetes. These are named based on the structures that they have when sexual reproduction occurs. So ascomycetes form an ascus or a sac. If you want to think of basically like a cigar, that's what an ascus is. And basidiomycetes, they form a basidium or a club. And typically ascomycetes have 8 spores and basidiomycetes have 4 spores when they reproduce sexually, but that's really not that important for this talk, but just in case you're wondering what the major differences are there. They're both eukaryotic organisms. They lack chlorophyll and they're members of the kingdom fungi, and they typically reproduce by spores and mycelia. Examples of ascomycetes in turf, so that would be the pathogens that cause dollar spot, your leaf spot, so gray leaf spot, and your Drexelera or bipolaris leaf spots. anthracnose, summer patch, necrotic ring spot, take-all patch, spring dead spot, pink snow mold. Those are all going to be examples of pathogens that are ascomycetes. Examples of basidiomycetes in turf would be things like the rhizoctonia diseases, so brown patch, large patch, yellow patch, whitea patch, red thread, gray snow mold, and fairy ring. Okay. So those are basically that's your general plant pathology that I wanted to give you a little bit of background with on, just so you all kind of get into today's discussion when we talk about controlling certain types of pathogens with DMI fungicides, and just to get some correct terminology out there that I'm going to use throughout the rest of the presentation. So now that we're going to move on to the fungicide portion of it, there's a few different things with fungicides that I wanted to cover again, just to give you all a review in case you haven't seen this for a while. So there's two types of fungicides. There's contact fungicides and there's penetrant fungicides. Contact fungicides, as the name implies, they're contact. They stay on the outside. They don't enter the plant. Examples of these in the turf industry would be chlorothalonil, fluazinam, and mancozeb. Coverage is critical because they don't enter the plant. So you want to have as much coverage on the leaf surface in order to prevent infection from occurring. Typically, these types of fungicides only provide 7 to 14 days of prevention from infection. And chlorothalonil and mancozeb have true multi-site activity and have a low risk for resistance. So you can think of this like a shotgun effect. It's hitting various parts of the metabolic pathway, which is really important when we begin talking about fungicide resistance. Penetrant fungicides, those enter the plant. They penetrate the plant surface. Examples are basically all your other groups of fungicides. So your DMIs, like we're going to talk about today, the strobies, the SDHIs, the QIIs, or the quinone inside inhibitors, the dicarboxamides, the polyoxins, the phenylpyrroles, and so on and so forth. Within the penetrant fungicides, you have three different types of movement or phytomobility. There's localized penetrants. So these basically, wherever the droplet lands, there's limited movement up. They're going to stay in the general vicinity. A lot of times they have translaminar activity, which means they'll go from one leaf side to the other to provide protection. There's acropital movement. Overwhelmingly, this is what most of your fungicides are within the penetrant fungicide group. They move up in the xylem. And then there are systemic fungicides, of which we only have one in the turf industry, and that would be aluminum tris, Fositoal, Chipco signature, however you want to call it. Penetrant fungicides have a site-specific mode of action, okay? So you can think when they go to take out the fungus, they have more of a rifle effect, right? They're hitting one specific spot. Because they're hitting one specific spot, penetrant fungicides are much higher at risk for resistance to develop to these types of fungicides. Now, one other thing I want to clarify or clear up, since we're talking about the types of fungicides, a lot of times people like to talk about, what fungicides have better knockdown control or have better quote unquote curative control of various pathogens. And to be honest with you, there really is no such thing. The way that penetrant fungicides work, so assuming you're going to make an application quote unquote curatively because you're seeing disease, right? What has to happen is the plant has to take up that fungicide first, okay? Then the pathogen that's residing in the plant that's causing disease has to also take up a fungitoxic amount of the chemical. Once that happens, you have really what you have is not fungicidal activity, but you're going to put the fungus into a fungistasis, meaning it's not going to grow anymore. It's basically in a dormant state. Okay, so when you make the application, not only does it have to get into the plant, it has to get into the pathogen and put it into fungistasis. And therefore, any curative effects that you may or may not see from fungicides are really because you're stopping the pathogenic attack and you're allowing the plant to recover. So I hate to be the bearer of bad news, but when you're looking for quote unquote curative properties, what you really need is to apply a fungicide and allow the plant, the infection to stop and allow the plant to recover. So this looks like we have some duplication here, but contact fungicides, as I said, don't enter the plant. I did want to mention they bind firmly to organic matter. which means there's limited to no control of root diseases. So if you're looking at contact fungicides like above, you don't typically want to use those for your root diseases. And a lot of times when you do go out with a quote unquote curative application, it's nice to go out with a contact because, as I said, fungi spread out radially and disease therefore spreads out radially. And what you can do is prevent plants at the peripheries from becoming infected and areas that are nearby from becoming infected by using a contact fungicide in conjunction with a penetrant should there be disease. Okay, we already went through this. Another important thing is that only 5 to 50% of penetrant fungicides are actually absorbed by the plant, and they don't stick around all that long. And because of that, since only this amount is taken up, and because it's usually gone within 14 days, that's why, because the fungus is exposed to it for such a long period of time and then allowed to grow back, that's why you need to reapply fungicides, but that's also why there's a higher risk for resistance to develop, because again, you're only going on after one part of the metabolic pathway. And right now, Lisa, we have a question for the participants. So the quick poll here is, what concerns you most about using DMI fungicides? Is it resistance? Is it regulation or phytotoxicity? Is it cost? Or is it spectrum of activity? Okay, Lisa, if you want to wrap up.
It's hard to sit when there's nothing happening, but I can see the votes coming in. So, yeah, closing the poll now. And there would be your results.
Okay, these are pretty interesting results. Fungicide resistance, obviously, that's part of the talk today, but it is a big problem with DMI fungicides. Regulation, phytotoxicity, that's another huge concern, which we'll get to. Spectrum of activity, we'll probably talk why this isn't as big of an issue with DMI fungicides. And then cost is kind of lower on the end, and that's usually pretty spot on with DMI fungicides, because the number of them out there, generally they tend to not be as costly as some of your other site-specific modes of action. So let's get to the actual portion of what we really wanted to talk about today, which is DMI fungicides. So what are DMI fungicides? They're broad spectrum control of ascomycetes and basidiomycetes, okay? Generally speaking, if you use a DMI fungicide, it's going to provide some level of control of almost every basidiomycete and ascomycete that we find in turf. They were first introduced in the 1970s, so they've been around for quite some time. Broadly speaking, they provide excellent control of pathogens that cause dollar spot, brown patch, summer patch, take-all patch, necrotic ring spot, and anthracnose in your leaf spots. Your ERI fungi, or your ectotrophic root infecting fungi, the patch diseases, these really are probably the best standalone active ingredients that there are. The triazole group of the DMI fungicides, they inhibit one enzyme, C14 demethylase, which really isn't that important for you to know, but it's critical for you to know because it's crucial for sterol development, okay? What are the challenges of DMI fungicides? Well, first is the obvious, particularly from some of the older PGR, or I'm sorry, some of the older DMIs, would be the PGR effects that we observe on turf. Next would be the potential for phytotoxicity, especially for those of you that work on warm season turf during the transition period. And there's a potential for resistance to develop due to overuse, particularly with pathogens with medium to high risk for resistance. So everybody is always concerned, especially in transition, with phytotoxicity from DMIs. And here was a study that we did a number of years ago at the University of Maryland. And some of these areas got significant phytotoxicity from the use of DMIs. And sometimes you see more of a regulation effect, like you see here. It's a little bit of a deeper blue. Now, if you're on a regular Primo, program, this may not be much of a problem to you. Of course, you always want to test DMIs with Primo or part of your program, just to make sure you're okay with any regulation that's occurring. And then you can get some phyto in certain instances. Typically, it's not going to be this bad, but this was a good slide to kind of show what you'd be looking for. So fungicide modes of action, right? What part of the cell are they hitting within a fungus? So if you think of this thing as a hyphae, okay, so it's a branch point of a fungal cell, you can see that you have different active ingredients or groups that hit different parts of the metabolic pathway. For our purpose today, what we're concerned with is the cell membrane, because that's the effect that DMI fungicides have to control fungi. This is actually taken from a practical guide to turfgrass fungicides. It was recently updated. It's a book by Rick Lattin. There's a lot of really good information in this book, a lot of graphical or pictorial representations like you see here, and it really gets into how fungicides work. So if you're ever looking for a good reference, that's a fantastic reference. The APS, or American Phytopathological Society, sells that book. So if you look at the DMI fungicides, right, they're part of FRAC group 3. They control sterile biosynthesis within fungal cell membranes. And the group that we particularly use in turf really only is going to be the triazoles. So examples there are difenconazole, flutriafol, methyltrifluconazole. Those are two of the newer ones. The metconazole, myclobutanil, and tebuconazole are again to be some of the older ones. But as you see here, and we'll talk about this reference later on, you can see that DMI fungicides have medium risk for resistance to develop. The active ingredients that you'd be familiar with, again, tebuconazole, that's torque, propiconazole, Banner Max, Balaton or triadimifon, difenconazole, which is really part of many premixes, Eagle or myclobutanil, and triticonazole or Trinity, and metconazole Turney. These were basically the standby DMI fungicides that we've had for the last 15 or 20 years or so that most people have been using if they've been using DMIs. Out of these single active ingredients, you have numerous premixed products. The list goes on and on and on. But here are just a few of those, right? And it's important, and I'm going to mention this again later, it's really important when you're developing your fungicide spray program, you really need to keep an eye on if you're spraying, let's say, tebuconazole or torque or any of the post-patent materials, right? You want to be careful not to go back out and start spraying Tekin for 2 apps or Traction for 2 apps or Fame right after that, or Fame plus T, I should say, because they all have tebuconazole. So when you are making applications or you're putting together your program, don't just think about spraying different products. You need to make sure that if you're spraying premixed products that they don't have the same group that you just sprayed 1 to 2 times previously, because that's really not resistance management, or at least the best practice of resistance management. There are a bunch of new DMI active ingredients to the market. When I say a bunch, I mean 3, but the way chemistry's been going lately, that's a fair amount of new active ingredients. So methyltrifluconazole, that's the first of these next generation DMIs that came out. That goes by the trade name Maxtima. It's made by BASF. So it has excellent turfgrass safety, including warm season turf during transition. Those of you working on Bermuda grass greens, if you are looking for a DMI to use and you've been afraid before because of the regulation or potential for phytotoxicity, this has really shown a lot of safety in transition. It has strong control over a variety of pathogens. One thing that Maxtima or methyltrifluconazole does not control would be Rhizoctonia solani, right? And that's, for those of you who have a lot of familiarity with the DMIs, it's kind of interesting because almost all DMIs are very strong on this particular pathogen, but not in the case of methyltrifluconazole. It's gotten very good spring dead spot control, but it only controls one of the 2 pathogens that causes spring dead spot. The other pathogen that control isn't as strong on as it is on Ophiosperella coriae. Flutriafol, this is Rayora. This is a new fungicide from FMC. Again, excellent turfgrass safety. This is kind of one of the hallmarks of these newer DMIs, including warm season turf grasses during transition. It's got strong control of a variety of pathogens. Just from stuff I've seen, there might be a little bit more extended control with Maxtima compared to Rayora. But again, Rayora provides very, very good control of things like your Rhizoctonia species. It's very strong with summer patch and also dollar spot. The newest one that's out, it's actually not a new DMI. Prothioconazole has been around for quite some time, but Bayer just put it out. The trade name is Densicor. I think it came out a few months ago. It's got excellent turfgrass safety, again, on warm season and transition. I saw it at a number of places on large patch this year. It performed very, very well. And again, just overall with dollar spot, brown patch, and your typical pathogens that you'd be looking for with the DMI, prothioconazole has done very, very well. So developing a program based on resistance management, how do you do that, right? So first, regardless of what class of chemistry you're talking about, whether it's a DMI fungicide or an SDHI fungicide or QOI fungicide, you want to make a list of what diseases affect your facility and when they occur frequently, okay? Are most of your problems occurring in the spring, summer, fall, winter? Okay, those who are most concerned with snow mold that are up in Maine may not have the same number of application issues with DMIs as those who live in the mid-Atlantic or Midwest. Next, know what active ingredients and or products are cornerstones for controlling the most problematic or hard to control diseases, right? So if summer patch is your biggest issue, okay, you need to know which AIs are the best on that. It's going to be your DMIs, your QOIs, and know which diseases are most at risk for resistance, right? So summer patch is really not a disease where you have to worry about resistance developing so much, okay? But other pathogens are, and we'll get to more about that here in a few slides. Once you have a foundation of products for the tough controlled diseases, surround them with the next best option, okay? But try to make it a different mode of action if possible. Rotating modes of action, as I said a few minutes ago, does not mean spraying different active ingredients from the same class. It means spraying active ingredients with different modes of action, okay? So spraying tebuconazole, and then your next spray 2 weeks later for dollar spot is propiconazole, and then your next application 2 weeks after that is prothioconazole, right? That's not resistance management or rotating. That's just using different active ingredients. So you want to switch your different classes or your different modes of action. Make sure you don't have more than 2 consecutive applications of site-specific modes of action, okay, at any time, right? So you can use 2 in a row, switch off to something else. If you have to come back to that mode of action again, fine, but make sure after 2 consecutive apps that you go to something else. This isn't something I made up. This comes from the Fungicide Resistance Action Committee. And remember that promoting resistance management results in achieving optimum disease control because different fungicide groups are going to remain efficacious against pathogens that are high at risk for resistance to develop for a much longer period of time, meaning you're going to get to keep using what you currently have instead of some of the behaviors that our industry has had in the past with getting a new mode of action or getting a new AI and kind of beating it into the ground and all of a sudden having issues with resistance. And remember, your go-to products for disease control, they're only as good as what you surround them with, right? So you always need to make sure that you're finding not only the best product out there, but the best 2, 3, or 4 products, especially ones from different groups or fungicide groups, so that you have the best programmatic approach you can to both control the disease, get the desired results that you want, and to be a good steward for resistance management. Just going to talk briefly here for a minute or two about Tekin fungicide, which is the DMI fungicide that we have at PBI-Gordon. It's actually a combination product, so it's a little bit of resistance management built in there. It's an SDHI fungicide and a DMI fungicide, so those are FRAC group 7 and 3. Because of the DMI, it's got really good broad spectrum control of ascomycetes and basidiomycetes. The SDHI component, isofetamide, really helps out with dollar spot and the spring dead spot pathogens. And we've also observed really fantastic control of summer patch with this product, as well as fairy ring and Whitea patch. So isofetamide's the SDHI, tebuconazole's the DMI. It has a very easy single use rate, 3 fluid ounces. Because of the TEB, you're limited to 3 applications of this product per year, and golf course use only. Fairways 21 to 28 day control of various pathogens is easily achieved with this product. And greens, I'd recommend 21 days, particularly if you're going back to back, just because you probably don't need that slug of TEB on a 14-day interval. But again, dual mode of action for a variety of different pathogens. Just to give you an idea, this is at University of Connecticut. Here's the non-treated check at this particular rating date. Here's Tekken on a 21-day interval and Tekken on a 28-day interval, controlling dollar spot really, really well. Here's data there just to show you comparatively to some of the other DMIs that are out there. Here's Tekken on a 21-day interval, Maxdema at the 0.4 fluid ounce rate on a 21-day interval, Banner Max at 1 fluid ounce on 21 days, and Tartan. And you can see Tekken has excellent control there with the other DMI fungicides, especially compared to the non-treated control. Here's anthracnose control. On the left, you have Tekken. This is on a 21-day interval. Again, in the center, you have Tartan Stress Guard. This particular location has some issues with QOI fungicides with anthracnose, so that's why you're seeing a lot more breakthrough with Tartan on this particular study. And here again is the non-treated check. You can see Tekken providing outstanding control of anthracnose. Brown patch, as you would expect with tebuconazole, fantastic control. This is 21-day interval. We typically see same results on 28-day intervals, again, compared to the non-treated check. And lastly, I think the hidden gem with Tekken, but again, this is with DMIs in general. They're really strong against summer patches. This is at Rutgers. Here's your non-treated on your left. Here's 3 applications, which you start when soil temps are 65 degrees to 2-inch depth on your right. And we've seen this year after year where it's almost 100% control. And here's data to really show that. And Tekken, again, compared to some of your other DMI fungicides, Maxdema, Rayora, and Fame plus T. And again, really good control. I know it doesn't look like 100% control in this graph, but this is really good control, especially as you get out this far from when applications were made. You're talking late August where this red bar is, and they're all providing really, really good control compared to your non-treated. So DMIs really need to be a part of your summer patch program, regardless of which one you use. They all bring something to the table for control of that. So go back to the phyto thing, just to kind of put some of you at ease. I kind of want to clear up maybe some misconceptions about the DMI use. You can see this type of regulation, as you see here, just a faint blue, again, similar to what you would see with Primo. I know that these yellow plots look very concerning, okay? So what I do is talk you through this. So we looked at a number of different things when we were developing our product Tekken, and we saw some pretty interesting results. So on the left-hand side, you can see this is tebuconazole by itself. This is not typical of what you see, but sometimes in the spring you can get a little bit of a shock. And this was after 1 application, okay? So you've got tebuconazole by itself, and then you have Tekken, okay, which includes the SDHI fungicide isofetamide. But you don't see any of that yellowing, right? Same place, here you see Balaton flow, okay, that's tridimifon. You see this yellowing again. When you add in the SDHI, you don't see it. So if you're concerned, sometimes looking at products that have an SDHI and a DMI, we believe there is some level of safening with this. Again, these aren't, you're not going to see this type of phyto all the time out of DMIs, but the weather conditions likely contributed to this a bit. But the addition of the SDHI in there, particularly with Tekken fungicide, we have seen a reduction in situations where you may see phyto. This graph basically shows you the same thing, showing that phytotoxicity, there was none, okay, with the non-treated. There really was none at all either with Tekken. There was some with Balaton, but I do want you to see here, these were applied every 7 days, and there were, I believe, 7 applications every 7 days to this turf, okay? So applying Tekken or tebuconazole every 7 days during the summer, we essentially saw no phyto at all on creeping bentgrass. So creeping bentgrass, if you're worried about DMIs, particularly Teb, don't be worried about that. If you get into POA and it's really, really stressed, that's a little different, but still you're more likely to see a little bit of the regulation than you are anything else. So just to kind of put that out there, if you want to do test plots, and if you're not using DMIs, but you really should be as part of a resistance management program, I wouldn't be as concerned with using them on creeping bentgrass and worrying about any type of phytotoxicity. Again, there may be some difference in the actual active ingredients you're using, but broadly speaking, especially with the newer DMIs, you don't need to worry, and they really need to be in there as part of your resistance management program. And Lisa, we have another question for the group here. All right. How many DMI applications do you make a year at your facility? 1 to 2, 2 to 4, 4 to 6, more than 6, or other? So we'll give you all a few minutes.
Brian, while they're voting on this, there's a question here that's come in.
Okay.
Is the SDHI stapling, or is it because a reduced rate AI in the mix?
No, so when I showed the two those two side by sides, the Tebustar and the Tekin, those were actually, as well as the triadimifon by itself and the triadimifon plus the isofenamide, those were equivalent amounts of both of those DMIs in there. So we do believe there was some safening. We had actually applied for a patent on that. The patent ultimately was not granted. But yeah, the amount of the DMIs in there, they were the exact same.
Okay, thank you. Here is what your poll answers look like. Okay.
So those are about on par. Depending, again, a lot of this depends on where you are. So if you're up in Maine, or you're in the Pacific Northwest, and your biggest issue is going to be microdochium or pink snow mold, and you don't need to get out there with too many fungicide apps during the year, I could see why 1 to 2 is normal. Anywhere from 4 to 6, I would say, is probably going to be what most people are doing. If you're able to use less than that in places where you see more or that have environmental conditions that are more conducive for disease to occur, that's probably where you're going to be. Using more than 6, you probably shouldn't be using more than 6. I would say 6 should probably be your max. Typically in resistance management, we've kind of looked at the industry. When I say the pathology industry has looked at typically using about 5 applications is where you should limit at-risk fungicides during, or at-risk fungicide classes in a particular growing season. Good. Appreciate you all participating in that. So we've been mentioning a lot about resistance, and I want to talk a little more about what that is. Okay, so fungicide resistance is a stable, inheritable adaptation of a pathogen to a fungicide, meaning it moves from the parent fungus to the offspring. Okay, and this also occurs in fungal-like organisms like oomycetes. Resistance of a pathogen may be caused by one gene, okay, or multiple genes. There are several mechanisms that cause resistance to develop, but most commonly in our industry, it's caused by target mutations. Okay, so the site that fungicide is going to go into to render the fungus no longer effective, that's a target mutation. Factors that influence the resistance include pathogen type, pathogen reproduction methods, pathogens with polycyclic cycles, right? So you're not going to expect something that has one disease cycle a year like summer patch or spring dead spot. Those pathogens aren't going to have a high risk for resistance, right? But your pathogens like Pythium, the pathogen that causes dollar spot, those are things that you're more likely to have a higher risk for resistance to develop. And also repeated applications from site-specific modes of action are going to be another big factor in how quickly the resistance develops or how widely it's seen. So once resistance is conferred, it's transferred to the offspring. And here are the mechanisms I was talking about. So if you think of the fungicide, if you look at this image on the right as these red triangles, right? These fungal cells or the target site looks like these little blue Pac-Man. Okay, so what can happen are a few different things. First, the fungicide goes into the fungal cell and it gets pumped out. Okay, you have modified efflux pumps. They basically just take it and pump it out. All right, this is the same. These are the same mechanisms of resistance that you have, like with any type of resistance, whether it's bacterial resistance or whatnot. including in humans. But the fungicide goes in, okay? The other thing that can happen is the target protein, the fungus starts to overproduce targets, okay? So there's just not enough of the fungicide present to stop fungal growth. The next would be the modified or mutated target site. Like I said, this is the most common one that we see. And then there's detoxification, which is a metabolic event where the fungal cell just basically breaks down the fungicide and doesn't allow it to do anything. And of course, in a perfect world, this is what you get every time you have the fungicide enter into the fungal cell, fit properly there into the target protein, and eventually it renders and puts that fungal cell into a state of fungistasis. Resistance development with the DMIs. So how does resistance develop? Okay, well, it's the same thing with any other class. It's by applying the same mode of action over and over and over with your at-risk pathogens. Resistance is specifically due to a mutation called CYP51, and this occurs at that 14 demethylase enzyme I was mentioning. It disrupts ergosterol formation. The 3 mutations that occur with the DMIs or with fungal cells to prevent the DMIs essentially to have resistance would be target site mutations. Okay, that's the most common. Modified efflux pumps. Okay, so the cells start to pump out the fungicide, and then target overproduction, where there's just more targets than there are molecules of the fungicide, and it's just rendered ineffective. What does this all mean in layman's terms? Okay, the cell membrane doesn't function properly and the cell dies. That's what happens when you apply a DMI and it works properly. But there are varying levels of insensitivity to DMI fungicides. It is important to know that cross resistance with the DMI fungicides does occur with the dollar spot pathogen. But in short, here's what this really looks like inside of a fungal cell. Okay, so here's your fungal cell, and the big concerns we talked about is disruption of ergosterol formation within the cell membrane. Okay, so ergosterol helps give structure to the cell membrane. When a fungicide works properly, what happens is the ergosterol is broken down, okay, and basically the cell kind of falls in upon itself. When you have resistance occurring, what happens is you make the fungicide application, okay, and it's going to break down the ergosterol, and you're inhibiting that ergosterol production, and you have these toxic sterols or just lack of sterols that don't give the membrane anything to stand on. And again, the cells that are proximal or that actually take up the fungus or the fungicide, they die, and nearby cells are put into fungistasis. So resistance occurs within a number of different groups. You have your aromatic hydrocarbons, your benzimidazoles, so thiophanate methyl. Everybody's familiar with thiophanate methyl resistance, particularly to the dollar spot pathogen. Your phenylamides, okay, so this would be mefenoxam. The dicarboxamides and phenylpyrols have resistance, so that's to an altered target site as well. You have your DMI fungicides, which have a number of different things, but I mentioned the 3 that we have in the turf pathogens, the efflux pump, the altered target site, and the target site overproduction. And then also your strobilurins have altered target site resistance. You notice these ones that just have the target site, that's where you're going to have what we call qualitative resistance, and that's going to be up here in another slide or two. So the pathogens that are most at risk for resistance that we see in turf are the Claridia species. We can't seem to find a good name for the dollar spot pathogens. If you're used to Sclerotinia homeocarpa, it's now either Claridia homeocarpa or Claridia jacksonii or Claridia monteithiana that we would see. Microdochium navali, that's the pathogen that causes pink snow mold or microdochium patch. Pythium aphanidermatium, which causes Pythium blight. Magnaporthia oryzae, that's the pathogen that causes gray leaf spot. And of course, Colletotricum cereale, that's your anthracnose pathogen. And these are the pathogens that are most at risk for development for resistance that we see in the turf industry. All right, I think we have one last poll question here.
We do have one last poll question, and it's on pathogens.
Yeah, which pathogens do you use DMIs for most frequently at your facility? We'll hope from what you learned earlier that it's not number one, but if it is, and you have used DMIs for Pythium, hopefully you won't going forward because of what you learned that the Pythium doesn't have ergosterol. But if you did, then you did, and you learned something new today, which is why we're here. Okay, Lisa, if you want to go ahead and end the poll. All right, good. So we did have a few in Pythium. Hopefully going forward, we won't use a DMI for that. But again, all really good pathogens, and that can be hard to control, particularly fairy ring and sometimes dollar spot. That's where you really want to use DMIs. Brown patch DMIs are really, really strong. The one thing I would say, and I do mention this a little bit later on in my slides, is that, you know, if brown patch isn't that hard to control, maybe conserve your DMI applications for things that are tougher, like dollar spot, fairy ring, summer patch, and things of that nature, take all patch. So going back to fungicide resistance, documented cases, okay? So here are the 5 groups, or I'm sorry, the 5 pathogens that I just mentioned. So the fungicide groups that show resistance to the dollar spot pathogens are the SDHIs, the DMIs, the benzimidazoles, and the dicarboxamides. For the pink snow mold pathogen, just the dicarboxamides for now. That there might be a change in that coming up here in the next year or so with some work we've seen. Pythium dermatum, so that's the phenylamides, so mefenoxam, as well as the strobilurin fungicides. Magnaporthia oryzae, which causes gray leaf spot, there's resistance with the strobilurin fungicides. Colletotrichum cereale, okay, so that's anthracnose, the DMI fungicides, the strobilurin fungicides, and the benzimidazoles, okay. So what you see here is that the toughest to control pathogens also happen to have resistance to the fungicides that we tend to use most frequently, the DMIs and the strobies, kind of across the board. Here are the groups on the bottom left of the screen that are most at risk for resistance. You got your benzimidazoles, the dicarboxamides, the phenylamides, the SDHIs, and the QOIs, okay? There's a really good graph in Rick Latton's book, A Practical Guide to Turfgrass Fungicide, I mentioned earlier. This is from 2011, but it's the same thing in his current book. If you look at the different pathogens, okay, so we just discussed that these are the pathogens most at risk biologically for resistance to develop, okay? On the other side, you look at the fungicide groups for their risk for resistance. And the neat thing that you can do with this is basically assess your risk for whatever you're using a fungicide for, okay? So for sake of today, we're talking about DMI resistance. If you're gonna use a DMI, right, you have a low risk for resistance if you're looking at summer patch, necrotic ring spot, spring dead spot, or fairy ring, okay? Resistance really isn't likely to develop from using DMIs for those pathogens. When you look at the combined risk moving up for pathogens like rust or leaf spot, you have an increased risk, right? But here really is what you want to avoid a lot of repeat applications for. That's using a DMI to control things like anthracnose, dollar spot, gray leaf spot, Pythium blight, and microdocium patch. That's not to say you shouldn't use them. That means that you need to be careful when using them, not to use more than 2 consecutive applications, and to make sure that you're rotating through other modes of action and adding in a contact fungicide wherever possible. So if we want to look at the different types of resistance, there's qualitative resistance. This is an all or none thing. This is a monogenic type of resistance, or meaning it involves one gene. So the benzimidazoles, the strobies, and the phenylamides, they have this type of resistance, okay? So what happens is you have a sensitive population, you keep making applications of the same mode of action over and over and over and over and over again, okay? Then all of a sudden you start to have, you know, an individual in that population that is no longer sensitive to the fungicide, right? Meaning there's a potential for resistance. Well, over time, you keep applying that same mode of action, and eventually you end up with a predominantly resistant biotype going on with your fungus, okay? And there's very few susceptible individuals left in that population. You have this over time, and what's going to happen is you're basically going to render fungicides from these groups ineffective, much like thiophanate methyl is for controlling dollar spot if you have issues at your facility. Quantitative resistance, so this is an amount, right? Not just a quality, it's an amount. There's varying degrees of what we would say is insensitivity. Examples of this would be your SDHI, DMIs, and dicarboxamides. So again, you're making applications of the same thing over and over and over again with your DMI fungicides. Over time, you start to have different individuals within that population with varying levels of sensitivity. And what you get if you keep applying those same fungicides over time is you may have a population that has some sensitive individuals, some that you may control 10 or 20% less than you normally did compared to the sensitive, and some that you may only control 50% as well as you did with the originally the sensitive individuals. So it's important to kind of understand not necessarily how these fully work, but just to know that with your different groups, there are two different types of resistance that occur, and that's because of the number of genes in the fungus that are allowing this or causing this to happen. But again, with DMIs, we're talking about quantitative resistance. If you look at resistance with the QOIs and benzimidazoles, again, this is qualitative. It's a quality. You either have it or you don't. You can see the frequency in the population shifts from low to high, and it either works or it doesn't, okay, with resistance. So there's no resistance over here, and there's resistance on this side. With your DMI fungicides, what you have after repeated applications is a shift to where you may see a little bit of a decrease in efficacy. You might even get to see some resistance, but we typically don't see populations where there's just complete resistance to a particular DMI fungicide, right? You have some level of insensitivity. I know these aren't DMIs here, but this is the best thing I can show you as an example of what quantitative resistance looks like. This is with SDHI fungicides. I'm sure many of you are aware that SDHI fungicide resistance with the dollar spot pathogen is readily happening and becoming more widespread. If you look here, there's 3 different mutations, okay, that occur with the SDHI fungicides on the dollar spot pathogen. And the sensitive population is right here. So in the sensitive population, the black box in all of these, okay, is your non-treated. So you're going to see lots of dollar spot in all of them, okay? The yellow box is boscalid. So in your sensitive population, boscalid works well. The blue box is fluxapyroxad or exemplar. The white box is isofenamide or cabuto fungicide. And the red box is fluopyram, which is part of Exteris. So in this one, the SDHIs all control the dollar spot pathogen. If you look at these different populations, right, you're going to see that in all of them, boscalid kind of gets beat up a little bit. You're going to see in 2 of them, fluxapyroxad gets beat up, but with this mutation, it's really not that bad. With isofenamide or cabuto, you're not bad here. You look really good here. It doesn't work on this population. And the same thing with fluopyram. It gets beat up a little with this mutation. Same here, but this one it's controlling. What this is trying to show to you is that with DMIs, it's really pathogen dependent. You might have different issues with different DMIs at your facility. You might get better control with tebuconazole than you do for mefentrifluconazole or vice versa. Again, it's very unique to your facility and the pathogens that you have there. One last look at this. Here's qualitative resistance in a Petri plate, okay? It's just occurring. This is a really high concentration, way above what you're using in the field. It's not stopping the fungus from growing. This would be your quantitative resistance right here. And you can see that when you get to field rates, you're controlling the pathogen compared to the non-treated. So that's what it looks like kind of in real life. Here's just a few more photos of that. Looking at those 2 populations, your non-treated and your SDHI fungicide, this is resistant, it's not doing much. Resistant population 2, here's your non-treated, here's your fungicide that's controlling it fairly well for the way that this trial was done with heavy inoculation methods. So how to effectively manage fungicide resistance, particularly DMI fungicide resistance? Again, be familiar with the different groups and what diseases they effectively control. Refer to FRAC. Rotate your modes of action, okay? No more than 2 consecutive applications, regardless of groups, but particularly with your DMIs. We don't have the same fungicide resistance issues with DMIs, like we've talked about, because of the quantitative resistance that you may see with the strobilurin fungicides. But still, you really want to make sure that we don't have widespread resistance to DMIs too. We're already seeing issues with the SDHIs. We know there's issues with the strobilurins, and we want to try to keep the DMIs from getting any worse off with different resistance to different pathogens than we already have. Always tank mix active ingredients with different modes of action if you can, especially multi-site, but be careful. If it's a pre-mixed product, you need to make sure that you're not spraying just different active ingredients from the same class repeatedly. And encourage turfgrass growth with fertility and BMPs. I know fertility on greens is not a great way that people like to control anthracnose, but if you want to see the best fungicide for anthracnose, just put down some quick release nitrogen, maybe 0.2 of a pound with a little potassium in it, and it'll be great. Best utilization of DMIs, remember, just because you're spraying it to control one pathogen, right? So let's say you're spraying to control Rhizoctonia solanae or brown patch. It doesn't mean that other pathogens aren't active, okay? So even though you're making your application to control brown patch or the pathogen that causes brown patch, the dollar spot pathogen might be active as well. And if you're making an application with your DMI to control Rhizoctonia the first week, and then 2 weeks later you're spraying another DMI to control the dollar spot pathogen, and then 2 weeks later you're spraying a DMI to control summer patch, well, you're just making 3 applications of DMIs in a row, which really isn't good. Look at for the pathogens that are most difficult to control that necessitate DMIs, right? Maybe use other classes for other pathogens where DMIs don't have to be used. That way you can save them and put them in your program where they're going to give you the most bang for your buck. Evaluate the resistance potential for control of pathogens when you're putting together this program with your DMIs. And again, go back to this. If you guys want to take a screenshot, I'll keep this up for a minute. If you want to take a shot with your cell phone, go ahead. Better yet, buy the book. There's a lot of really good information in there on all this stuff, on diseases, on practices, on all kinds of interpreting fungicide data. Anyway, this is a good tool to look at your potential for resistance to develop. Another good one is to go to the Fungicide Resistance Action Committee page. Just type in FRAC or FRAC worksheet or FRAC codes into Google. This will come up, and every year they put this out, and they have all the different modes of action. They talk about the group name, what they actually do, the different common names that you'll see in there, resistance risk, and gives you the codes too. So it's a really, really good reference. It's only about 4 or 5 pages. Another great reference is this joint publication put out annually by Kentucky, Rutgers, and Wisconsin. It's called Chemical Control of Turfgrass Diseases. And if you look at that, it's also available as a handout with this today. But if you go through for each different disease or pathogen, they talk about the different fungicides that are available, efficacy, and a lot of times they have notes like this where it says 1/3. That's depending on whether or not there's resistance. The intervals tested, and it's basically one big publication put out. It's about 20 pages long with information from fungicide trials across the country. Most importantly, with DMI resistance management, right, there's lots of single active ingredient DMIs that are out there right now, okay? There's also a lot of pre-mixed products that have DMIs out there right now. A lot of these are great fungicides. They need to be part of your fungicide program to properly control various pathogens, but you need to make sure that if you are making applications, you're not just using products that have DMIs in them over and over and over again, because what that's going to do is render DMIs less effective for controlling the problematic pathogens at your facility. And overall, you're not being a steward to the industry if you're not willing to go a little further to make sure that you're properly rotating through your groups to prevent DMI resistance from occurring. In summary, resistance of pathogens, it's actively occurring and evolving. It's always going on. It's just kind of how biology works. Resistance is a stable and heritable adaptation of a pest to a pesticide. Resistance can be monogenic or polygenic. Obviously, the monogenic is much more problematic because you just have complete loss of those fungicides. DMIs are quantitative, but again, don't even let it get to that point where you're starting to lose efficacy. Major pathogens of cool season grass tend to be much more, have much more issues with resistance than you do with pathogens that are problematic on warm season turf, with the exception, of course, of dollar spot and Pythium blight. It's imperative to alternate your modes of action and use BMPs wherever possible. Incorporate multi-site fungicides wherever you can, as often as you can, into that resistance management program. And new technology and combination products, they afford turf grass managers with an ability to combat resistance. But like I said in the last slide, just make sure that you're not overusing those premixed products to where you're just making repeated applications from the same group. I appreciate all of you listening today. I do a lot of tweeting with regards to product development or weather patterns, things that are out there. I tweet about turf and only about turf, or travel related to turf. So if you want to follow me on Twitter, I try to put out a lot of helpful information out there for superintendents to use. And because we put this event together and you all were gracious with your time to listen to me, you can go ahead and use the code at the bottom here, 999-238-27-31525, to get your education points. And with that, I appreciate you all listening.
Brian, you even gave them the code. Nice.
I know. I figured I'd help you out.
I appreciate that. That's wonderful.
Are there any questions I can answer while you're all taking note of that number?
Let's see if anybody has their hand raised. Want to raise your hand? I can unmute you, or you can type into the question answer box.
And just because I work for PBI-Gordon, if you have questions with any products, go ahead and ask them. I did a lot of work in my graduate years with products across the board, and I'm happy to share with you things that have worked well, and things that you know, I'd point you towards if you have specific questions.
That's very kind. We've got a quiet group today, I guess.
All right, well, you all have my email here and my Twitter handle. So if you ever want to send me a DM or an email with questions you have, or you're working on a program and you want to tell me what the pathogens are that you have issues with and want me to look it over, please feel free. Or if you see me at a conference, stop me, introduce yourself, and I'll be more than happy to chat with you.
Okay, let's see if they're going to. Not seeing anything further right this time, Brian. I guess you answered all their questions as we went along.
Yeah.
I will say that we had planned to have a third episode in the series, and unfortunately our instructor had to cancel. So we are going to, so save the date, October 20th will be the next PBI-Gordon webinar. All the details on that will be forthcoming. In case you missed the first, Brian taught on Pythium disease management last month, and that is available on demand in the Learning Hub on the website. If you go to the GCSA website, you'll see that there. Maybe you'll do that when you're inputting your code for your education points there. For those people who are listening in the future, use the date you're listening, not our original presentation date of today. I want to thank Brian for teaching today. I want to thank PBI-Gordon for their support of this and other GCSA services. I guess if we have no other questions, we have a couple of different people who are saying thank you, and it's appreciated. So
thank you all for your time. I appreciate it, and being good listeners. And again, if there's anything I can ever help any of you with, please don't hesitate to reach out. More than happy to help.
Thanks, Brian. I know that's appreciated. And with that, we'll call it a day. Everyone stay well. We'll see you online again soon. Bye now.